membrane separates the outer cellular environment from the inner cellular environment. The ions are not equally distributed across the membrane. The cell is abundant in potassium and anions such as amino acids, but sodium chloride and calcium are present in higher concentration extracellularly. The voltage difference across the cell membrane at rest is known as resting membrane potential. It is present in all the cells. It is around minus 70 millivolt to minus 90 millivolts. Now we'll talk about the sodium potassium ATPase pump. It is electrogenic pumps. Electrogenic pumps are those pumps which work across the membrane. During its function, it produces electrical imbalance. The sodium potassium ATPase pump is present in all the cells. It brings 2 potassium inside the cell but transfers 3 sodium outside the cell using ATP. As it keeps pumping, cations move extracellularly. The cells becomes negative. All the cells are neutral. As cations are binded with anions, but when the sodium potassium pump works, it takes 3 cations, leaving 3 anions, but brings only 2 cations intracellularly, so it produces negativity inside the cell. Another important channel which is present in the cell is ungated potassium channels or leaky potassium channels. There are basically 3 types of the channels: ungated channels, which are also known as leaky channels; voltage gated channels, which are active at specific voltage; and ligand gated channels, which when the ligand is bind to the receptor, they activate. So the ungated potassium channels or leaky channels allow the potassium to move across the cell membrane. A lateral electronegative force, which is of minus 5 millivolt, which is produced by the sodium potassium ATPase pump, holds back the potassium to move extracellularly, but the concentration gradient, which is greater than the electronegative force, so the potassium leaks extracellularly. As the potassium moves extracellularly, leaving the anions behind, it will produce negativity in the cell. This negativity produced due to diffusion of the potassium is called potassium diffusion potential. As more and more potassium gets out, the potassium diffusion potential increases, and a time comes when potassium diffusion potential becomes equal to the concentration gradient. When potassium diffusion potential becomes strong enough to oppose the concentration gradient, there is no net movement across the membrane of the potassium, in spite of the fact that the membrane is still permeable to potassium. At that time, membrane has achieved the potential which is called potassium equilibrium potential. Equilibrium potential for any ion is the Nernst potential for that ion. The Nernst potential is another name of equilibrium potential. When there is concentration gradient difference across the membrane and membrane becomes permeable to the ion due to its concentration gradient difference, that ion will move across the membrane and produce electrical potential. That electrical potential is produced by the diffusion of the ion, so it is called the diffusion potential. A time comes when the diffusion potential becomes so strong that it opposes the concentration gradient, which results in the stoppage of the net movement of the ions, in spite of the fact that the membrane is still permeable to that ion. At that point, membrane has achieved equilibrium potential for that ion, or the Nernst potential for that ion. Equilibrium potential for potassium is diffusion potential of potassium at which net movement of potassium is zero in spite of the fact that the membrane is fully permeable to potassium. equilibrium potential for potassium is Nernst potential for the potassium, which is around minus 85 millivolt. Resting membrane potential produced. In all human cells, there are a lot of sodium potassium ATPase pump, which are concentrating sodium out and potassium in. So all the cells are very rich in potassium. Resting cells are not permeable to sodium, but they are very permeable to potassium. So all the human resting cells diffuse little bit of potassium to develop enough diffusion potential of potassium. So further potassium will not leak out, and that happens to be around minus 80 millivolt. So human resting cells are allowed to achieve Nernst potential for potassium when they are at rest. This potassium efflux out of the cell, which normally occurs in the all resting cells, is mainly responsible for resting membrane potential. The role of sodium potassium ATPase pump in our resting membrane potential direct contribution is of electronegativity of minus 5 millivolt. But the indirect role is the major role, that is to maintain high concentration of the sodium extracellularly and high concentration of potassium intracellularly. So the potassium leaks out and resting membrane potential is achieved. In resting cells, the inner side of the membrane is polarized negatively. Nernst equation is the equilibrium potential for one ion. It is E is equals to minus 2.3 into R into T upon Z into F into log of 10 concentration inside the cell upon concentration outside the cell. E is the Nernst potential or the equilibrium potential. This is the constant. The R is the gas constant, and T is the temperature, and F is the Faraday's constant, and Z is the charge of the ion. So if we cancel out all the constants, this minus 2.3 gas constant, temperature, and this Faraday's constant, so it will all sum into minus 60 millivolt upon the charge of the ion into log of 10 into concentration inside the cell upon concentration outside the cell. So the Nernst potential for the potassium is minus 85 millivolt. For sodium, it's plus 60 millivolt, and chloride, it's minus 85 millivolt. And the Goldman equation is basically the sum of the Nernst potential for all these ions. It is electromotive force is equal to minus 60 millivolt. This minus 60 millivolt into potassium concentration inside the cell upon potassium concentration outside the cell, and permeability of potassium, and this permeability of potassium plus sodium concentration inside the cell and sodium concentration outside the cell, permeability of sodium and permeability of sodium. This whole thing will be is equal to 0 as the cell are impermeable to the sodium at rest. And the chloride concentration inside the cell plus chloride concentration of the cell outside the cell, and the permeability of the chloride. This is basically sum of the ions, sum of the Nernst potential for each ion, which is known as the Goldman equation. It's just the sum of the Nernst equation of potassium, sodium, and chloride.